Welcome. This is our new channel which is specifically for Olympiads. This is the first lecture in this channel. As you have seen, we will be discussing regarding jumping genes or what we call transposons. Regarding that, first of all, we would start with a process, rather a special topic. Say we will start with bacterial genetics. We will start with bacterial genetics. In the very beginning, I would like to say that for those who have completed the part of Kingdom Monera from NCERT, as well as Cell the Unit of Life Prokaryotic Cell part from NCERT, can watch this lecture. Well, if you have those parts remaining, I don't think that would be very much helpful to go through this lecture. So first, we will start with the part of bacterial genetics, and eventually we will understand the part of transposons or jumping genes. First, regarding bacterial genetics, simply if we look into a bacterial cell, say this is a bacterial cell, and we know this cell do not have any nucleus. There is a chromosomal DNA or the main DNA present in the cell, and there might be some other extra chromosomal DNA which we call plasmids. So say this is the DNA, or we can say the chromosomal DNA. So simply when bacteria divides, it just produces two daughter cells. Prior to fission, this process is, we know, it is known as fission. And prior to this process, they replicate their DNA. So once there was a question, I can't actually recall the year presently. This question asks, when does bacteria divides its DNA, or when does replication take place in a bacterial DNA? So if this process is binary fission, here is one DNA, but after that fission we have two DNA. So must be before this process of binary fission, bacteria might have created another copy of this DNA, and eventually one have entered into this cell and the other has incorporated into this cell. This should have occurred. So if the question says when does DNA replication take place in a bacterial cell, the answer would be easier, that prior to fission या फिर before binary fission, the DNA of bacteria replicates and eventually gets transferred into two daughter cells. So simply this seems like mitosis, but actually we use the term here binary fission. But there is a problem in this process. In this process, the main problem that would arise is there would be no genetic variability. Now, what does the term genetic variability actually signify? Here, since this is a type of asexual reproduction, you might know that binary fission is actually a type of asexual reproduction. In such kind of processes, there is no genetic variability, or there is no change in the DNA sequence of parent as well as daughter cell. These two daughter cells have exactly same type of DNA which actually resembles the parent DNA. So there is no change or no variation, which was the main power for evolution of sexual reproduction, for which or you can say another type of cell division actually occurred, that is meiosis. That is, the evolution of meiosis actually took place when one bacterial cell that initially used to divide by asexual reproduction did not show any genetic variability. And if in the process of evolution higher organisms or advanced organisms would evolve, genetic variability would be a major force. So in this type of process there is no genetic variability. So the question arises, does bacteria can actually show any kind of genetic variability? The answer is yes. Bacteria can show genetic variability. So simply we can say genetic variability also occurs in bacteria. This can also occur in bacteria, but this is definitely not by fission. Not by fission. Such kind of genetic variability in bacteria is not definitely by the process of fission, because in fission we can say that there is no genetic variability. So what would be the process called? This process would be, actually there are 4 types, yeah, we can say 4 processes by which bacteria can show genetic variability. So genetic variability in bacteria, it can simply take by 4 processes. The first is what we call conjugation. The second is known as transformation. The third one, transduction, and the fourth one, transposition. Now see, there is a slight catch in this topic. We are not studying regarding sexual reproduction in bacteria. If the question says sexual reproduction in bacteria takes place by, this could be either of the first 3 processes, that is conjugation, transformation, or transduction. We are talking about genetic variability, how bacteria can show any sort of genetic variability. So there are 4 processes. I am discussing this process, that is transposition. I will be dealing with this process called transposition, how transposition actually occurs. So before I actually start explaining you what is transposon, what is jumping gene, I would like to elaborate regarding the process of transposition, because this entire process produces a protein or an enzyme. You can say this is transposon. So how or where we will study this process, this entire part was connecting part. The actual lecture will start from here regarding the topic, as I say, transpositions of or transposons. So before we start what is transposon or transposition, we need to know where from this term came. This term came regarding the reproduction in bacteria. In classical binary fission, we do not see any genetic variability, but bacteria can also show some genetic variability by 4 processes. The first process can be conjugation, second can be transformation, third transduction, and fourth transposition. Now we will talk regarding this process. I will elaborate this process for you. So first, what is transposition? I would actually elaborate it in a different manner so that it would be easier for you to understand. First, say, let us consider this is a DNA sequence. This is a DNA sequence. Here, there is a gene. This part actually is a gene. Is it visible? Yes. This part is a gene. Now, this gene can, you can say, this gene, it can jump or change its position from here to this part. Is this color visible? Okay. This gene can jump or change its position from here to this part. So the resulting sequence would look something like this. This changing of position is called as transposition. Now I will get a bit deeper into this topic. The same diagram in a different way. Say this is a bacterial DNA. Chromosomal DNA. Although it's circular, I am showing it in a linear fashion to make you understand it better. And in this DNA, we are concerned regarding, say, this gene. specifically this sequence. Now I am telling you that this sequence is actually a transposition sequence. That means it can change its position. This sequence can change its position from somewhere here to say here. And if it changes the position, it would simply look something like this. This gene has actually changed its position from here to here. This is known as transposition. This process is actually what we call transposition. I hope I'm clear with this part. Transposition, that is changing of position of a gene on a DNA. This is simply called transposition. Now from this diagram, I will show you the types. This is a type of transposition. Another type of transposition could something be like, say this is a DNA, and here is a gene. This gene will now change its position, say from here to this part. It will change its position from here to this part, but here, before changing its position, it would create its replica, or it would create a copy here, and then it would replicate another sequence there. So something would something can be seen like, say here initially, here was a sequence. Now this sequence here actually made a copy here, and another copy was sent there. Say this is another copy of the same sequence. So in the first case that we saw, in this case there was one copy, and this actually changed its position. There was one gene that actually changed its position. But in the second process, this one copy made its another replica, and that replica was sent there. So these are the two types of transposition. In this transposition, this part of gene did not replicate. This gene did not replicate and just changed its position. Whereas in the second type, this gene made its replica. One copy was left out here, and the other replica was sent in a different position on the DNA. Here, since there was no replication, this type of transposition is known as non-replicatory transposition. This type of transposition is known as non-replicatory transposition. Why? What could be the possible reason? Because the name itself suggests, because this gene did not replicate here, and this entire gene was altered from its position or changed its position. Whereas in the second case, we can see that this gene made a replica here, and another copy was generated which was transferred to a distant location on the same DNA. This type of transposition is known as replicatory transposition. I hope I am clear with this. Actually, it would be better, or it would be rather easier, I could say, that you can remember it. Say here, this entire sequence was transferred from here to here. We can say, or we can remember it by cut plus paste. I hope you all know what is copy paste and what is cut paste. So we can remember it by cut and paste, and here we can remember it by copy paste. I don't think that I need to elaborate this part further. I hope it is clear what is replicatory and non-replicatory transposition. Next, significance. What is the significance of this change? So before we deal with the actual part called significance, I would like to show you how actually this jumping of the gene takes place. Say this is a bacteria. This bacteria has its DNA. This is a chromosomal DNA, and say it has another set of DNA which we call plasmid. So simply here, this is a chromosomal DNA. This is a chromosomal DNA, and this one is an extra chromosomal DNA or plasmid. Means this is not the main DNA, extra chromosomal. That means in some bacteria this DNA may be present or may not be present, because this is not the main DNA or the chromosomal DNA. This is an extra chromosomal DNA called plasmid which may be present in some bacteria. Now let's check what actually happens here. Say this bacteria has a sequence, and this sequence here in this chromosomal DNA, I'm just checking out this part. Say I'm only seeing this part, only this part of the sequence. Clear? I'm just making it a bit larger. So this is that part of the sequence. This entire sequence we call it transposable element. This entire sequence we call it a transposable element. In this transposable element there are two parts, one the end parts and one the middle part. This end parts here, this end parts, this sequence and this sequence, these are present at the ends. These sequences are called inverted repeats. These sequences are called inverted repeats, one here and the other here. This one would also be called inverted repeat. And that middle part, this entire part, this sequence is called insertion sequence. This one would be called an insertion sequence. Now what actually happens? I hope you can recall this part. Now you see, say this is the entire sequence of the circular DNA. This is the entire sequence of the circular DNA. And somewhere here, say this part, this sequence was present. Somewhere here in this part, this sequence was present. what is this sequence? This sequence is actually, here we have inverted repeat, here we have inverted repeat, here we have insertion gene or insertion sequence, and this entire one is what we called a transposable element. This transposable element can change its position and enter into some other part. Say it changes its position and enters here. This sequence where it actually enters. Here, this entire sequence, I hope it is clear, this entire sequence will now come here. It may create a copy and replicate and come here. It might not create a copy and this entire sequence will be cut and come here, as I told you in cut paste or copy paste. I said this entire sequence might come directly here, or it might be that it created a copy here and then it will jump there. That means one copy will remain here and the other copy will go there. If it does not create any copy, that one we call non-replicatory transposition, and if it creates a copy here and sends another copy in somewhere else or in a distant area, it will be called replicatory transposition. Now this sequence, or say this area where this sequence will enter, this area where this sequence will go, this is called the target sequence. Let me show this entire thing in a big cell of bacteria. It will be more helpful. This part is very important to understand this actual process. This is a bacterial chromosomal DNA, and this one here is a plasmid DNA. Say here this part is actually a transposable DNA. That means a transposable element. I am telling you, this will change its position. This may actually change its position from here to here. Say it will enter in this part. This will be called transposition. It also can change its position from here to here. This one will also be called as transposition. Now is that clear? What actually transposition is? There is a gene sequence. This one. This one is called a transposable element. This transposable element can change its position here in the same chromosome in some other part. It can also change its position to a nearby plasmid. Even if this cell is infected by a bacteriophage virus, it may incorporate this sequence into that bacteriophage sequence. So these are all the possibilities. But here we are not dealing regarding the virus. This sequence, we named it transposable element. This sequence we named it transposable element. This might be readable. Yes, this sequence is called transposable element. This transposable element here on the chromosomal DNA can shift its position somewhere here into the same chromosomal DNA or into the plasmid. This entire process, either it can take place here or it can take place here. Either way, it would be called this would be called transposition. Now I hope it is clear what actually transposition is. I have still not made you explain regarding the transposons. I am just talking regarding the transposition. Transposition means this sequence will change its position either on the same DNA or on a different plasmid. Now I will talk regarding transposons. Say this was the sequence. This was the entire sequence and this was the transposable element. This transposable element actually This is a segment of DNA. This part will form an RNA. This will form an RNA. And eventually, this process is called what? Transcription. And this RNA will now translate into a protein. This RNA will now translate into a protein, or you can say a polypeptide. This protein can actually act as enzymes like transposons. These proteins can actually act as enzymes and are called transposons. Since they are able to change position, this transposable element gene can change its position. They are also called jumping genes. Jumping genes, why? Because they can change their position in the same chromosome or in a different plasmid. That is why this is known as jumping gene. And ultimately, a protein is produced, that is transposons. Now, what is actually the significance of this part? This entire process, shifting of a sequence from here to here, or shifting of a sequence from here to here, why would this be actually necessary? The main necessity of this process would be like, say this is a Enterococcus bacteria. This is a bacteria. It's named as Enterococcus. And I hope you already know, whenever this term arises, entero, this somewhat refers to the intestine. Means this is a bacteria which is present in the intestine. Now, if a bacteria is present on intestine, I would definitely take some antibiotic to kill this bacteria. If my intestine has a bacteria, I would definitely take some antibiotics because this bacteria is harmful, or you can say pathogenic. So the name of the antibiotic that we use here to kill this bacteria is vancomycin. Okay, let me write it for you. The name of this antibiotic is vancomycin. This vancomycin is an antibiotic that we use to kill this bacteria. But there is a problem. Bacteria would itself try to evolve some mechanism through which it can get resistant to these antibiotic. From my point of view, I would try to kill this bacteria. From bacterial point of view, it would try to resist the vancomycin action. So if vancomycin is coming, it would try to resist the vancomycin action. How? By transposition. There is a transposable gene. Here, there is a transposable gene. This transposable gene has its name Van-A gene. This transposable gene, that is, it can change its position, it can do transposition. This transposable gene can actually alter the peptidoglycan sequence. Now this part might seem to be a bit confusing. Peptidoglycan sequence. Now what does that actually mean, peptidoglycan sequence? Say this is a bacteria. This bacterial cell wall, say this one is a bacteria. This bacterial cell wall is, this outer layer is cell wall. The cell wall is made up of peptidoglycan, or what we commonly call murin. Now this peptidoglycan has a specific sequence. That's why I initially said that the part of Monera in biological classification and the part of prokaryotic cell in cell the unit of life should be very clear. Here you will get to know that the peptidoglycan sequences are actually made up of repeating units of NAG, NAM and some pentaglycine bridges. So now we will not go deep into this structure. I am simply telling that let us consider this is a bacteria. This outer covering is called a cell wall. This outer covering of the bacteria is called a cell wall. Now this cell wall itself is made up of peptidoglycan, and this peptidoglycan has a specific sequence What this transposon gene or when a gene does is that it changes this sequence. Say initially this sequence was A, B, C, D, E. And what does vancomycin do? Say I'm telling you, initially, say the peptidoglycan is what? Peptidoglycan is the cell wall of bacteria. Say initially the peptidoglycan sequence was something like A, B, C, D, E. This was the cell wall of bacteria, the sequence. What vancomycin actually does, say vancomycin actually breaks this part A and B. Wherever it will find the sequences A, B, it will make a cut here. As a result, if the cell wall is made up of A, B, C, D, E, and vancomycin antibiotic disrupts the A and B sequence, this entire cell wall will break and the bacteria will eventually die. But what this VAN A gene does, this VAN A gene actually reverses the sequence. Say the sequence is now something like this. Now if vancomycin comes and this sequence is altered, this is altered by VAN A. If this sequence is altered, then whenever vancomycin will come, it originally attacks the AB sequence, but here there is no AB sequence. After A there is C, before B there is D, so there is no AB sequences here. So vancomycin cannot kill this bacteria. What actually took place? See, initially this was the bacteria and this was its DNA, and its cell wall, this part is called what? The cell wall, which is made up of peptidoglycan. When vancomycin comes, it attacks on the cell wall sequence. But when this VAN A gene got active, say somewhere here is a VAN A gene. When this gene got active, this altered the peptidoglycan sequence. Now when vancomycin was actually given, it could not actually bind to the cell wall part, because initially it would have attacked the Enterococcus bacteria, but since the sequence of cell wall is altered, it cannot find a target site which it can bind and affect. So if you think from bacterial point of view, this bacteria has developed vancomycin resistance. Means what will actually happen? I am having Enterococcus in my intestine, I am taking vancomycin. I would expect that the vancomycin would kill the bacteria, but it actually cannot kill this bacteria, because bacteria has developed such a mechanism, or it has such a transposition of VAN A gene that it altered the sequence of the bacterial cell wall, making it resistant to vancomycin. This vancomycin resistance will lead to failure of antibiotic action. Means I will take that antibiotic, but the bacteria will not be killed, or there will be very poor efficacy. The patient having Enterococcus will not be cured by vancomycin if it develops resistance, if the Enterococcus bacteria develops resistance. How does it develop resistance? By altering the peptidoglycan sequence. Why peptidoglycan sequence alters? Due to this VAN A gene. What is actually a VAN A gene? This is a transposable element. Due to transposition, actually this VAN A gene changes the peptidoglycan sequence, because of which this vancomycin cannot identify the peptidoglycan part. Clear? So simply what will happen, due to this altered sequence, now vancomycin, which is an antibiotic, won't act against this Enterococcus. Means the patient will take antibiotic, but the bacteria will not be killed, and this is what we call resistance, antibiotic resistance, or here the name of this antibiotic is vancomycin. we would name this bacteria as vancomycin resistant enterococci or enterococcus. Is it clear? This bacteria will now be called as vancomycin resistant enterococcus, or simply we will denote it as VRE. I hope this process is clear. What is vancomycin resistance? Now, what could be another significance of this? I will just see entire part in an extended manner. So, there would be a quick recall itself. Say this is an enterococcus. This is an enterococcus. Clear? Now this enterococcus has Van A gene, which ultimately led to vancomycin resistant enterococcus. Clear? Vancomycin resistant enterococcus. We saw it just now. Now what happens? Say this is a colony of different bacteria. These bacteria are called as Staphylococcus aureus. These bacteria are called as Staphylococcus aureus. Now what actually happens? This enterococcus has a Van A gene. Say this is an enterococcus. Sorry. This is an enterococcus having this one. This one is a Van A gene. This one up here is a Van A gene. Now what actually this enterococcus does? It sends this Van A gene into the Staphylococcus colony. This Van A gene is transferred. Say this is a Van A gene. This is a Van A gene. It is transferred into the Staphylococcus colony. As a result of which Staphylococcus aureus now has a Van A gene in itself. This one also has a Van A gene, and we know when there was a Van A gene, in enterococcus, we saw vancomycin resistant enterococcus. When there was a Van A gene in the enterococcus became vancomycin resistant. Now if the same gene is present in Staphylococcus, now we will find that this Staphylococcus also becomes resistant to vancomycin. We will call it vancomycin resistant Staphylococcus. Is that part clear? Means this gene can be transferred from here to here, as a result of which what would be its result? Staphylococcus would be vancomycin resistant. Staphylococcus becomes vancomycin resistant. So if we used to call this one vancomycin resistant enterococci, we can also say this one as vancomycin resistant streptococcus. Is that part clear? Why bacteria needs this part is what we call significance. Why bacteria does transposition? This is what we study under the topic of significance. Why? An enterococcus has a gene called Van A gene, which is a transposable element. This transposable element can or has the potential to make it vancomycin resistance. If this Enterococcus becomes vancomycin resistant, we call it vancomycin resistant Enterococcus. Similarly, if this Van A gene gets transferred into a colony of Staphylococcus, this Staphylococcus also does the same. What? It will alter the peptidoglycan sequence of the bacteria. And ultimately, vancomycin also can't act on this Staphylococcus colony, as a result of which we will see vancomycin resistant Staphylococcus. Now a question might arise in your mind, how is this process actually possible to transfer this Van A gene from here to here? This can be made possible either by conjugation, as we initially said, the three processes. Is the ink visible? No. Let me change the color. This process is made visible. This process is made possible either by the process of conjugation or transformation or transduction. I hope it is clear now. This Enterococcus having the Van A gene becomes resistant, and we call it vancomycin resistant Enterococcus. We can also transfer this Van A gene from here to a Staphylococcus colony. Eventually this will also become resistant to vancomycin, and we call it vancomycin resistant Staphylococcus aureus. I hope this entire part of transposition was clear. You got to know what is transposon, what is a jumping gene. I hope that part is clear. With that, I would like to conclude this first video on this new channel of Faction Olympiads. Regularly I would try to make some videos because I also have to take some offline classes. I will try to post some videos regularly that are in this channel. Specifically, I would like to focus on such videos which would be either a bit out of NCERT or a very elaborated version of NCERT. We will solve some PYQs of NSEB and INBO in some short lectures, hardly 5 to 10 minutes for each question we will take. We will solve 1 or 2 questions in a one lecture or one video, and that would be from NSEB or INBO 2019-22, these parts accordingly. So I hope you understood this lecture, but as I said earlier, this would be beneficial if you would have already studied those parts regarding the structure of bacterial cell. Okay, so in the next lecture I would try to elaborate these topics: conjugation, transformation, transduction a bit. Here we will see regarding F plus F minus, as well as high frequency of recombination conjugation, HFR positive and recombination negative part in conjugation. Second, in transformation we will see how a bacteria takes a DNA from the extracellular matrix, or we will see transduction in two processes. Transduction is of two types: a general transduction and a specialized transduction, both of which we will look. Clear? So let's conclude it here. Thank you.